Hello, everyone. I'm Elizabeth Block, and my senior thesis is on the analysis of pyrite nodules within central Pennsylvania bentonite. My advisors are Dr. Heaney and Dr. Gold. So these pyrite nodules are interesting because they're not found in surrounding bentonite beds. I'm looking into why they were formed, how they were formed, and when they were formed relative to the deposition of the host bentonite. So the outline of this presentation is I'm going to be going over my hypothesis and the overview, field images, methods, data analysis, my conclusions, steps for further research, acknowledgements, and references. My hypothesis is that the pyrite nodules found within the bentonite beds in central Pennsylvania were formed as concretions within the bentonite beds following their deposition. Bentonite is volcanic ash that has been altered after deposition into clay minerals as a result of weathering or subaqueous alteration. So here's one of my field sites in Agony Quarry. The bentonite beds are very well exposed at quarries. So the red arrows are pointing to the two bentonite beds here, two prominent ones. And the bentonite beds that I am studying were deposited from volcanic eruptions into the ocean around 450 million years ago. They are found among middle and upper Ordovician limestone beds. And you'll notice there is some orange oxidation coming down from this upper layer of bentonite. This is indicating that there are likely pyrite nodules in that bentonite layer because the iron in the pyrite is oxidizing. So my two collection sites were Oak Hall Quarry and Nagany Quarry. Here's what the pyrite nodules look like in place within the bentonite. So on the left, you can see these nodules sort of found together, and then on the right, these are some of the most spherical nodules that I found. And you'll notice that all of the nodules have a slight white coloration. Well, we'll get back to this later. So there are some samples in the field that showed how the pyrite nodules evolve. So this is over here in the left, you can see these tan streaks here and here. These are actually pyrite nodules. They're just early evolutionary stages where they're composed of a majority of host bentonite clay and not as much pyrite. So they're elongate. They don't have a shape or form as opposed to most of the nodules collected like here and here, but this nodule is also an early evolutionary stage. So I measured the density of the nodules in order to gain more insight into their composition and the percentage of host bentonite clay versus pyrite in them. So to measure their densities, I used this triple beam balance. This setup, there was a string coming down into this bucket of water. And so what I did was I measured the mass of each nodule in the air in this tray here. Then I submerged it in water for 5 minutes. Then I weighed it again. I performed a simple calculation and got these results for densities. So as you can see, this is density versus the amount of samples that had that particular density. Most of the nodules fell within 4.1 to 4.5 grams per centimeters cubed. Now this makes sense because the density of pure pyrite is 5 grams per centimeter cubed. So it makes sense that since they have some of the host bentonite clay incorporated in them, they're just below 5 grams per centimeter cubed. However, there is one outlier over here at 2.8 centimeters per cubed. This could likely be an early evolutionary nodule. So I used two main methods for my analysis of both the pyrite nodules and the host bentonite. I used X-ray diffraction, XRD, along with scanning electron microscopy, SEM, and electron dispersive spectroscopy, EDS. To prepare my pyrite nodules for XRD, I transformed them into a powder. Did the same thing with the bentonite. And then for SEM, I transformed the pyrite nodules into thin sections, and I used the same bentonite powder that I used for XRD for SEM. I sprinkled it onto this tape and stub here. I also coated the thin section with carbon, and that is carbon tape right there. So here is my pyrite nodule X-ray diffraction data. So I used Jade to analyze the data and characterize the sample. So I found that it contains quartz, pyrite, calcite, and montmorillonite. The montmorillonite is from the host bentonite clay that's incorporated in the nodule. So this dark blue line here is the original scan. So the bentonite X-ray diffraction data tells us that there is quartz, montmorillonite, and actiganite. Here is an SEM and EDS bentonite data. So on the left, we have an electron image, and you'll notice this shape in the center. This is the characteristic shape of a gypsum crystal. And gypsum is composed of calcium and sulfur, so it makes sense that the calcium and sulfur elemental maps show the nanoparticles of calcium and sulfur condensed in the area where this crystal is. So gypsum likely formed as an evaporated brine solution in the water table, went through the bentonite as the water table went up and down, and as the water table fluctuated, the bentonite became wet and then dry, wet and dry. And as this was happening, the calcium and sulfur likely were accumulating and agglomerating to form the gypsum. The pyrite nodules that I showed you in the field earlier had that slight white coloration. This could possibly be the gypsum from the bentonite. Now, the pyrite nodules likely formed at a completely different time than the gypsum and are not related at all to their formation, but the iron and sulfur nanoparticles likely accumulated in a similar way as water infiltrated through the bentonite. You'll notice that the iron and sulfur particles are concentrated in some of the same areas. So here is an SEM image and some elemental maps of a pyrite nodule. And the white background, the matrix here, is composed of iron and sulfur, so it's the pyrite. And then the dark parts of the image are clay blebs from the host bentonite. So you'll notice that the cubic structure of pyrite here is impinging on the clay bleb. And you can see the cubic structure in other parts of the image as well. This arrow here is pointing to this light gray bleb in the string of clay blebs. Not sure what this is. It could be calcium carbonate because there is a little bit of oxygen in the elemental map there. But the lack of concentric circles and radially fibrous crystals is important here because concretions, those are characteristics of how concretions grow. So they could just not be visible because it's too high of a magnification, but they're also not visible in the samples if you're just looking at them in your hand. So this is telling us that the preferred terminology for these pyrite nodules is likely nodules over concretions. So another way to get the density of the nodules is by using the SEM images of the pyrite nodules and using ImageJ software. So what the ImageJ software did was for each image, it separated the light parts of the image, which was pyrite, from the dark, which was bentonite, and it gave the percentage of each. And so from this percentage, I was able to perform calculations to find the density of the nodule as well as the percentage of pyrite versus clay. So over here for this image, I got a density of 4.97, and over here for this image, I got a density of 4.74 grams per centimeter cubed. These are higher densities than measured by hand, and they also contain a higher percentage of pyrite than measured by hand. So in conclusion, the pyrite nodules are still best referred to as nodules rather than concretions because of the lack of concentric circles in the SEM images. The XRD data reveals nanoparticles of iron and sulfur that likely aggregated to form the pyrite nodules with the aid of water infiltrating through the host bentonite. And the pyrite nodules formed following the deposition of the bentonite due to the incorporation of bentonite clay within the nodules. So some steps for future analysis would be the use of a light microscope to collect photos of the nodules, an iron isotope analysis, the use of transmission electron microscopy for analysis at a higher magnification. This would allow us to obtain images of the individual pyrite crystals that could be useful, a microprobe analysis of the limestone, and the analysis of more pyrite nodule thin sections, as well as more areas within each thin section. Thank you to my advisors, Dr. Peter Heaney and Dr. David Gold, for their time, effort, knowledge, and enthusiasm. Thank you to Dr. Maureen Feynman for guidance. Thank you to all the laboratory technicians that helped me with operating the laboratory instruments. Thank you so much.